Hi everybody, we are going to move on now and we're going to start to talk about cell communication. So, how do cells communicate? With cellular phones. Seriously though, cells actually do need to be able to communicate with each other for several different reasons. One is regulation. Cells need to be able to regulate or control what's happening within their boundaries, but they also need to be able to respond to things that are happening outside of their boundaries. So they need to be able to react to their environment. We are going to look at two general types of cell signaling. The first is local signaling, and so that's going to be, an example would be synaptic signaling, so how nerve cells communicate with each other. And then long distance signaling, the good example is the endocrine system, and so how hormones are released from endocrine cells. They'll travel through your bloodstream and then end up at a target cell. And then those signals will have an effect on the target cell. There are three general stages of cell signaling that we're going to look at. The first is reception, so how the signal is actually received. Next is transduction, and so how that signal is passed on within the cell itself. And then response, so what is the cellular change that's happening because of the signal that was received. So reception, here we have the signal molecule. That is in the extracellular fluid. This gray area is the plasma membrane. You can see we have a receptor protein, so this is an integral protein within that plasma membrane. And then over here we have the cytoplasm, or the inside of the cell. And so the signal molecule is going to bind to the receptor protein. After that, that receptor protein is going to pass on the signal, so this is transduction, and so it starts a series of events within the cell. And then finally you have the response, or that activation of something within the cell, some type of response. So again, reception is the target cell's detection of a signal coming from outside the cell. This can happen through direct contact or through signal molecules. And direct contact is when molecules flow directly from cell to cell without actually crossing a membrane. So this has to do with some of the stuff we talked about earlier in the week. With plants, you have the plasmodesmata, and then with animals, you have those gap junctions. And so signals are just going to flow from one cell to the other through these cellular openings. This can also occur by the cell surface molecules that project from the surface, and they're actually going to touch another cell. And so this is the example down here. You have cell-to-cell recognition. So you've got molecules on the outsides of the cells that interact with each other, and then that's going to start a signal being sent within this cell over here. So signal molecules are really important. It's the actual chemical that's going to travel from cell to cell. Now, a lot of times these molecules can't cross cell membranes, and the reason for that is because they're usually water soluble. So again, they're not going to like that hydrophobic region of the phospholipid bilayer, and they're also usually pretty large, and so they're going to be too large to actually cross through the membranes. And so because of that, they behave as ligands, or a small molecule that's going to bind to a larger one. The larger molecule that these signals are binding to are usually going to be made of protein, and they're going to change shape when they're bound to a signal molecule, and that's what's going to usually start this signal. And so the signal is going to transmit information from the exterior to the interior of the cell. There are several different receptor mechanisms that we're going to take a look at in this chapter. G-protein linked, tyrosine kinase ion channels, and then intracellular communication. And today we're going to focus on the G-protein linked receptor mechanism. And so this is going to be a plasma membrane receptor. And so it works with G-protein, which is an intracellular protein with a GDP or a GTP molecule. So here, again, we have our extracellular fluid. And here is the cytoplasm, and so this is our plasma membrane. We are looking here at this receptor molecule, so it is a protein. It has a signal binding site on the outside, and then it's got a segment that's going to interact with the G protein on the inside of the cell. So this G protein is going to use GDP and GTP as a switch. And so the GDP is the guanosine diphosphate, GTP guanosine triphosphate, and so just like ATP and ADP, GDP is going to be inactive. It has less energy than GTP. That is the active form. And so when it's active, the protein is going to bind to another protein, and it's going to alter its activation. And so this active state is only temporary, so it very much acts as a switch where it's turned on only when that switch is on, and then it gets turned off. All right, so it should make a little bit more sense right now. So here's the system in its inactive form. So again, extracellular fluid is blue, cytoplasm is the yellow part, and then this gray is our cell membrane. And so this is the G protein linked receptor right here. You can see it's got the reception site for the signal molecule. There currently is not a signal molecule, so it's inactive. This is our G protein, and it's got a GDP bound to it, so again, inactive. And down here is an enzyme. Now, when the signal molecule binds to the G protein linked receptor, this receptor is going to become activated. The GDP is going to become displaced by GTP. Then this activated G protein is then going to travel down the cell membrane. It's going to link up with this enzyme and activate it, and then this activated enzyme is then going to cause some cellular response. When all of this is over, this phosphate is let go, and so now we have the GDP again on the G protein. It's going to travel back, and you can see here that this signal molecule has released. And so we're returning to the inactive form. So G protein linked receptors are really important. They have very diverse functions. So things like vision and smell, very important for that. A lot of diseases actually work by affecting G protein linked receptors. So things like whooping cough, botulism, cholera, even some cancers can affect the way that G protein linked receptors work. And this number kind of varies. Sometimes you'll see up to 60%, sometimes you see slightly lower estimations. But either way, lots of medicines exert their effects through these G protein linked receptors, so very important in the pharmaceutical and medical fields. So your lecture question for tonight is, what do you think would happen if G proteins were unable to convert that GDP back to GDP? All right, so send me a quick note with your answers, your thoughts on this, and I'll see you guys soon. Thanks.